and genetic erosion. Classify the gene pool of a crop. The gene pool refers to the collection of all the genes and their different alleles within a population or species. It encompasses the genetic diversity present in a population, including the variations that determine different traits and characteristics. The gene pool concept is important because it influences the adaptability and resilience of a population to environmental changes, as well as the potential for evolutionary processes to occur. Genetic erosion, on the other hand, refers to the loss of genetic diversity within a population or species over time. It occurs when certain alleles or genes are lost from the gene pool, resulting in reduced genetic variation. Genetic erosion can be caused by various factors, including natural selection, genetic drift, and human activities such as habitat destruction, overexploitation, and selective breeding. When classifying the gene pool of a crop, we typically consider the following categories. 1. Wild relatives. This includes the genetic diversity present in wild plants closely related to the cultivated crop. Wild relatives often possess valuable traits that can be utilized for crop improvement, such as disease resistance or tolerance to environmental stress. 2. Landraces. These are traditional varieties of the crop that have been developed and maintained by farmers over generations. Landraces often exhibit high genetic diversity and adaptability to local conditions. 3. Improved varieties. These are cultivated varieties that have been developed through breeding programs or genetic engineering. Improved varieties usually possess specific traits or characteristics that make them desirable for commercial production, but they may have reduced genetic diversity compared to landraces. 4. Modern cultivars. These are the commercially dominant varieties that are widely cultivated. Modern cultivars often have been selected for high yield, uniformity, and other desirable traits. However, they may have limited genetic diversity due to the focus on specific traits during breeding. It's important to preserve and maintain the genetic diversity within the gene pool of a crop to ensure its long-term sustainability, adaptability, and resistance to pests, diseases, and environmental changes. Conservation efforts, such as the establishment of gene banks and the promotion of on-farm conservation, can help prevent genetic erosion and protect the gene pool of crops. Describe the advantages and disadvantages of mass selection, simple recurrent selection and clonal selection. Mass selection. Advantages. 1. Simplicity. Mass selection is a straightforward breeding method that does not require complex procedures or extensive resources. 2. Genetic diversity. It allows for the preservation of a relatively high level of genetic diversity within a population since it involves selecting desirable individuals based on their phenotypic characteristics. 3. Adaptability. Mass selection can lead to the adaptation of a population to local environmental conditions over time, as the selection is based on the performance of individuals in their specific environment. Disadvantages. 1. Slow genetic improvement. The genetic progress achieved through mass selection is typically slower compared to other breeding methods that involve more sophisticated techniques like molecular markers or hybridization. 2. Lack of precision. Mass selection does not consider individual genetic merit and can result in the retention of undesirable genes along with desirable ones. 3. Limited trait control. It is challenging to select for complex traits or those influenced by multiple genes using mass selection alone. Simple recurrent selection. Advantages. 1. Increased genetic gain. Simple recurrent selection allows for faster genetic improvement compared to mass selection. By selectively breeding superior individuals and then crossbreeding their progeny, it facilitates the combination of favorable genes from different individuals. 2. Trait improvement. It allows for simultaneous selection for multiple traits, including those influenced by multiple genes, by carefully designing the breeding program. 3. Genetic diversity. While simple recurrent selection involves some level of inbreeding, it still maintains a higher genetic diversity compared to clonal selection. Disadvantages. 1. Complexity. Simple recurrent selection requires more planning and resources compared to mass selection. It involves multiple steps, including crossing, selection, and backcrossing, which may require careful management and record keeping. 2. Inbreeding depression. The repeated rounds of inbreeding involved in recurrent selection can lead to a decline in performance due to the expression of deleterious recessive alleles. 3. Time and labor intensive. The implementation of recurrent selection programs requires significant time and effort for the development and evaluation of breeding populations. Clonal selection. Advantages. 1. Genetic stability. Clonal selection ensures the preservation of desirable traits and genetic stability over generations since the offspring are genetically identical to the parent plant. 2. Rapid multiplication. Clonal propagation allows for the rapid production of large quantities of genetically uniform plants, ensuring consistent and predictable crop performance. 3. Trait fixation. Clonal selection is effective for traits that are controlled by a few major genes and do not exhibit high genetic variability. Disadvantages. 1. Limited genetic diversity. Clonal selection can result in reduced genetic diversity, as it involves the propagation of genetically identical plants. 2. Vulnerability to diseases and pests. If the selected clone is susceptible to a particular disease or pest, the entire crop can be at risk due to the lack of genetic variation and diversity. 3. Lack of adaptability. Clonal selection may lead to reduced adaptability to changing environmental conditions since the genetic uniformity limits the ability to respond to new challenges or stressors. It's important to note that these advantages and disadvantages may vary depending on the specific crop, breeding objectives, and selection criteria employed. Different breeding methods can be combined or tailored to address specific breeding goals and challenges. Explain the importance of somatic hybridization in agriculture. Somatic hybridization is a technique used in agriculture to combine desirable traits from two different plant species or varieties into a single hybrid plant. It involves the fusion of protoplasts, cell components without cell walls, from different parent plants, resulting in a hybrid with a combination of traits not found in either parent. The importance of somatic hybridization in agriculture is as follows. 1. Genetic variation. Somatic hybridization increases genetic variation by introducing new genetic material into the crop species. This can lead to the development of plants with improved traits such as disease resistance, increased yield, enhanced nutritional content, and tolerance to environmental stresses. 2. Crop improvement. By combining desirable traits from different plant species or varieties, somatic hybridization allows for the rapid creation of novel and improved crop varieties. This technique can accelerate the breeding process by bypassing the limitations of traditional sexual hybridization, which may be hindered by genetic barriers or incompatibilities between species. 3. Disease resistance. Somatic hybridization can be utilized to transfer disease resistance genes from wild or resistant species into commercially cultivated crops. This helps in developing crop varieties with improved resistance to various pests, pathogens, and diseases, reducing the reliance on chemical pesticides and enhancing crop productivity and sustainability. 4. Abiotic stress tolerance. Somatic hybridization offers a means to introduce genes that confer tolerance to environmental stresses such as drought, salinity, extreme temperatures, or nutrient deficiencies. By incorporating these traits into crop plants, somatic hybridization can help mitigate the negative impacts of adverse environmental conditions, ensuring stable and productive crop yields. 5. Crop diversification. Somatic hybridization facilitates the expansion of crop diversity by enabling the combination of traits from different species or varieties. This diversification can help address the challenges of monocropping, such as increased vulnerability to pests, diseases, and environmental fluctuations. It also provides options for the development of new crops or crop varieties with unique traits or characteristics. 6. Conservation of germplasm. Somatic hybridization can be used for the conservation of endangered or rare plant species by fusing their protoplasts with those of related species. This approach helps to preserve the genetic diversity and unique characteristics of endangered plants, which can be challenging to conserve through traditional breeding methods. Overall, somatic hybridization plays a crucial role in agricultural research and crop improvement by expanding the genetic diversity, enhancing desirable traits, and addressing the challenges faced by modern agriculture. It offers a powerful tool for developing resilient and sustainable crop varieties to meet the increasing demands of food production and mitigate the impacts of changing environmental conditions. Intellectual property rights, IPR, issue is a sensitive topic in agriculture. Concisely describe this with your conclusions. Intellectual property rights, IPR, in agriculture refer to the legal protections and rights granted to individuals or organizations for their innovative plant varieties, genetic materials, and agricultural technologies. This includes patents, plant breeders' rights, trademarks, and other forms of IPR. The issue of IPR in agriculture is sensitive and complex due to several reasons. 1. Access to genetic resources. IPR can create barriers to accessing genetic resources, particularly for farmers, researchers, and breeders in developing countries. Patents on plant genes or varieties can restrict the use of these resources, potentially limiting the development of new crop varieties or hindering research. 2. Control over agricultural inputs. IPR can grant exclusive control over agricultural inputs such as seeds, leading to the concentration of power and market dominance by a few large corporations. This can reduce competition, limit choice, and increase the dependence of farmers on a limited number of patented varieties or technologies. 3. Impacts on farmers' rights. IPR can potentially undermine farmers' rights to save, reuse, and exchange seeds, which have been integral to traditional farming practices. Restrictions on seed saving and sharing can impact small-scale farmers' autonomy, self-sufficiency, and their ability to adapt to local conditions. 4. Impact on food security and biodiversity. The commodification of seeds and plant genetic resources through IPR can prioritize commercial interests over the broader goals of food security and biodiversity conservation. Concentration of IPR in the hands of a few entities may result in the loss of traditional and locally adapted varieties, reducing agrobiodiversity. 5. Biopiracy concerns. IPR can be used to claim ownership over traditional knowledge and genetic resources that have been developed or conserved by indigenous and local communities for generations. This raises concerns about biopiracy, where valuable traditional knowledge is exploited without adequate benefit sharing with the communities involved. In conclusion, while IPR can incentivize innovation and investment in agricultural research and development, it also raises significant challenges and potential negative impacts in the context of agriculture. Balancing IPR protection with broader considerations of access, equity, biodiversity, and farmers' rights is crucial. It requires thoughtful policy frameworks, fair benefit sharing mechanisms, and a recognition of the diverse needs and contributions of different stakeholders in the agricultural sector. Asexual reproduction. Asexual reproduction is a mode of reproduction in which offspring are produced from a single parent, without the involvement of gametes or the fusion of genetic material from two individuals. It results in offspring that are genetically identical or very similar to the parent. Here are some key points about asexual reproduction. 1. Lack of genetic variation. Asexual reproduction does not involve the recombination of genetic material, leading to offspring that are genetically identical or clones of the parent. This lack of genetic variation can limit the ability of the population to adapt to changing environments or overcome challenges such as diseases or predators. 2. Efficiency and rapid reproduction. Asexual reproduction can be highly efficient and rapid, allowing for the production of numerous offspring within a short period. This can lead to a quick expansion of the population and colonization of new habitats. 3. Preservation of desirable traits. Asexual reproduction is advantageous when a parent possesses highly favorable traits, as those traits are directly passed on to the offspring without the risk of dilution through recombination. 4. Mitosis and budding. Asexual reproduction can occur through various mechanisms, such as mitosis, where the parent cell divides to produce genetically identical daughter cells, and budding, where a small outgrowth or bud develops from the parent organism and eventually separates to form a new individual. 5. Regeneration and fragmentation. Some organisms can reproduce asexually through regeneration, where lost or damaged body parts are regenerated into complete organisms. Fragmentation is another form of asexual reproduction, where an organism breaks into multiple fragments, each of which can develop into a new individual. 6. Examples of asexual reproduction. Asexual reproduction is common in many organisms, including bacteria, archaea, some plants, e.g., through vegetative propagation or runners, some animals, e.g., parthenogenesis in insects and certain reptiles, and some fungi, e.g., through spore formation. Asexual reproduction offers advantages such as efficiency, rapid population growth, and the preservation of desirable traits. However, the lack of genetic variation can limit adaptability and increase vulnerability to environmental changes. Sexual reproduction, which involves the recombination of genetic material from two parents, provides genetic diversity that enables populations to evolve and adapt more effectively. Both forms of reproduction have their unique advantages and are employed by different organisms based on their ecological and evolutionary requirements. 2. Progeny test. A progeny test is a breeding technique used to evaluate the genetic worth or performance of an individual by examining the characteristics of its offspring. It is commonly employed in agriculture and animal breeding to assess the hereditary qualities of breeding stock. Here are some key points about progeny testing. 1. Purpose. The primary purpose of a progeny test is to estimate the genetic value or breeding potential of an individual based on the performance of its progeny. It provides valuable information for selecting superior parents for future breeding programs. 2. Controlled breeding. Progeny tests are typically conducted under controlled breeding conditions to ensure accurate assessment of the parent's genetic influence. Controlled matings are arranged to produce offspring that can be compared and evaluated based on specific traits of interest. 3. Phenotypic evaluation. Progeny testing involves the evaluation of offspring based on various phenotypic traits, such as growth rate, yield, disease resistance, or other desirable characteristics. These traits are selected based on the breeding objectives and goals of the breeding program. 4. Statistical analysis. Progeny test results are analyzed statistically to estimate the breeding values or genetic potential of the parent being tested. Statistical methods, such as heritability estimates, can help quantify the genetic contribution to the observed traits. 5. Sample size and replication. To ensure reliable and accurate results, progeny tests require sufficient sample size and replication. Multiple offspring from each parent are evaluated to account for genetic variation and to minimize the influence of random factors. 6. Long-term evaluation. Progeny tests are often conducted over several generations to evaluate the stability and consistency of the parent's genetic performance. Long-term testing helps in identifying genetic trends, assessing genotype-environment interactions, and validating the breeding value estimates. 7. Application in plant and animal breeding. Progeny testing is applied in various fields, including crop improvement, livestock breeding, and forestry. It helps in the selection and identification of superior individuals for further breeding, ensuring genetic progress and the development of improved varieties or breeds. Progeny testing plays a crucial role in genetic improvement programs by providing objective and empirical data on the genetic potential of individuals. It enables breeders to make decisions and select the best candidates for future breeding, leading to the development of improved, productive, and resilient populations. 3. Seed bank. A seed bank, also known as a gene bank or seed vault, is a facility or organization that preserves and stores a collection of seeds for future use. It serves as a repository of genetic diversity and plays a vital role in the conservation and protection of plant genetic resources. Here are some key points about seed banks. 1. Preservation of genetic diversity. The primary purpose of a seed bank is to conserve and preserve the genetic diversity of plant species. It collects and stores seeds from a wide range of plant varieties, landraces, and wild relatives, ensuring their availability for future generations. 2. Conservation of endangered and rare species. Seed banks often focus on collecting and storing seeds from endangered, rare, or locally adapted plant species that are at risk of extinction. By safeguarding their genetic material, seed banks contribute to the conservation of biodiversity and the protection of ecologically important plant species. 3. Protection against catastrophes. Seed banks act as a form of insurance against natural disasters, climate change, or other catastrophic events that could lead to the loss of plant species or their genetic diversity. The stored seeds provide a backup in case of seed loss in the wild or during times of environmental stress. 4. Research and plant breeding. Seed banks serve as valuable resources for scientific research, plant breeding, and the development of improved crop varieties. The stored seeds can be utilized to study genetic traits, adaptability, and potential applications in agricultural and horticultural practices. 5. Exchange and access. Many seed banks facilitate the exchange of seeds among researchers, breeders, and farmers, promoting collaboration and sharing of genetic resources. Access to the seed collections is often governed by international agreements and protocols to ensure equitable and fair use. 6. Long-term storage. Seeds in seed banks are stored under controlled conditions of temperature, humidity, and light to maintain their viability for an extended period. Some seeds can remain viable for decades or even centuries, allowing for long-term preservation of genetic material. 7. International seed banks. Notable seed banks include the Svalbard Global Seed Vault in Norway, the Millennium Seed Bank project in the United Kingdom, and various national and regional seed banks around the world. These seed banks work together to create a global network for seed conservation and exchange. Seed banks are crucial for safeguarding plant genetic resources, promoting biodiversity conservation, and supporting agricultural and ecological research. They provide a valuable resource for future generations, ensuring the availability of diverse genetic material for crop improvement, environmental restoration, and addressing the challenges posed by changing climates and emerging agricultural needs. 4. Genetically modified crop. Genetically modified crops, GMCs, also known as genetically engineered, GE, crops or biotech crops, are plants that have been modified through genetic engineering techniques to introduce specific traits or characteristics. These modifications involve the insertion or manipulation of genes from different organisms into the crop's DNA. Here are some key points about genetically modified crops. 1. Traits and benefits. GMCs are developed to possess specific desirable traits that can provide various benefits, such as improved pest and disease resistance, herbicide tolerance, enhanced nutritional content, increased yield potential, and tolerance to abiotic stresses like drought or salinity. 2. Genetic engineering techniques. The genetic modification of crops involves techniques such as gene insertion, gene editing, e.g., CRISPR-Cas9, or gene silencing, RNA interference. These techniques allow for precise manipulation of the crop's genetic material to achieve the desired traits. 3. Enhanced agricultural productivity. GMCs are developed to enhance agricultural productivity by reducing crop losses due to pests, diseases, or weeds. Traits like insect resistance can reduce the need for chemical insecticides, while herbicide tolerance allows for more effective weed control, potentially increasing yields and reducing production costs. 4. Food security and nutrition. GMCs have the potential to contribute to global food security by increasing crop productivity and reducing post-harvest losses. They can also be engineered to improve nutritional content, such as enhancing vitamin or mineral levels in staple crops to address micronutrient deficiencies. 5. Environmental considerations. GMCs have the potential to reduce the environmental impact of agriculture. For example, insect-resistant crops can reduce the need for chemical insecticides, benefiting both the environment and human health. However, the potential impacts of GMCs on non-target organisms and ecosystems require careful evaluation and risk assessment. 6. Regulatory oversight. GMCs are subject to strict regulatory oversight in many countries to ensure their safety for human health and the environment. Regulations typically involve assessment of potential risks, mandatory labeling requirements, and rigorous testing before commercial release. 7. Public perception and debate. GMCs have been the subject of public debate and differing opinions regarding their safety, environmental impact, ethical considerations, and potential socio-economic consequences. These discussions often revolve around issues of consumer choice, labeling transparency, intellectual property rights, and the role of large agribusiness corporations. It's important to note that perspectives on genetically modified crops can vary, and opinions range from support for their potential benefits to concerns about potential risks and unintended consequences. The continued development and adoption of GMCs involve ongoing scientific research, responsible governance, and open dialogue to ensure the safe and sustainable use of this technology in agriculture. Define cell. How is a plant cell different from an animal cell? Describe a typical cell structurally and functionally, with a suitable diagram. A cell is the basic structural and functional unit of all living organisms. It is the smallest independently functioning unit of life, capable of carrying out essential biological processes. Cells can vary in size, shape, and specialized functions depending on the organism and tissue type. Plant cell versus animal cell. While both plant cells and animal cells share many similarities, there are some key differences between them. Plant cell. 1. Cell wall. Plant cells have a rigid cell wall made primarily of cellulose. This provides structural support and protection for the cell, giving it a fixed shape. 2. Chloroplasts. Plant cells contain chloroplasts, which are responsible for photosynthesis, the process by which plants convert sunlight into chemical energy. 3. Large vacuole. Plant cells typically have a large central vacuole that stores water, nutrients, and waste products. It helps maintain cell turgidity and provides support to the plant structure. 4. Plasmodesmata. Plant cells are connected by plasmodesmata, small channels that allow for the transport of nutrients, water, and signaling molecules between adjacent cells. Animal cell. 1. No cell wall. Animal cells lack a rigid cell wall, providing them with greater flexibility in shape and movement. 2. No chloroplasts. Animal cells do not possess chloroplasts, as they are unable to carry out photosynthesis. Animals obtain energy by consuming other organisms or organic matter. 3. Small vacuoles. Animal cells may contain smaller vacuoles, but their size and number can vary depending on the cell type and function. 4. Gap junctions. Animal cells are connected by gap junctions, specialized protein channels that allow for direct communication and the exchange of small molecules between cells. Typical cell structure and function. A typical cell consists of various components that work together to carry out essential functions. Here is a brief description of the main structures and their functions. 1. Cell membrane. Plasma membrane. The cell membrane is a semi-permeable barrier that encloses the cell, controlling the movement of substances in and out of the cell. 2. Nucleus. The nucleus contains the genetic material, DNA, of the cell, which controls cell activities and stores instructions for protein synthesis. 3. Cytoplasm. The cytoplasm refers to the gel-like substance inside the cell that surrounds the organelles. It serves as a medium for cellular metabolism and supports organelles. 4. Endoplasmic reticulum. The ER is a network of membranes involved in protein synthesis, lipid metabolism, and the transport of materials within the cell. 5. Golgi apparatus. The Golgi apparatus modifies, sorts, and packages proteins and lipids for transport to their final destinations inside or outside the cell. 6. Mitochondria. Mitochondria are responsible for cellular respiration, producing energy, ATP, through the breakdown of glucose and other nutrients. 7. Ribosomes. Ribosomes are the sites of protein synthesis, translating the genetic code from the nucleus into functional proteins. 8. Lysosomes. Lysosomes contain digestive enzymes that break down waste materials, cellular debris, and engulfed pathogens. 9. Cytoskeleton. The cytoskeleton provides structural support to the cell and is involved in cell movement, cell division, and maintaining cell shape. Here is a simplified diagram illustrating the structural components of a typical animal cell. YAML copy code _____________ vertical bar nucleus vertical bar __________ vertical bar vertical bar vertical bar mitochondria vertical bar endoplasmic reticulum Golgi apparatus ribosomes lysosomes This diagram represents a basic animal cell structure, emphasizing some of the key organelles and structures. Keep in mind that the actual structure of a cell can vary depending on its specific type and function within an organism. Describe the physiological and molecular basis of heterosis. Heterosis, also known as hybrid vigor, refers to the phenomenon where the offspring of two genetically distinct parents exhibit superior traits compared to their parents. It is commonly observed in hybrid plants and animals. The physiological and molecular basis of heterosis can be attributed to several factors. 1. Complementation of dominant alleles. Heterosis can result from the combination of dominant alleles from different parents. If one parent has a dominant allele for a specific trait, and the other parent has a different dominant allele for the same trait, the offspring can inherit both dominant alleles, resulting in enhanced expression of the trait. 2. Overdominance or heterozygote advantage. Heterosis can also be explained by overdominance, where the heterozygous state, having two different alleles, at certain gene loci leads to superior performance. In heterozygotes, the interaction between the two alleles can result in improved physiological processes, such as increased growth, disease resistance, or reproductive fitness. 3. Genetic complementation. Heterosis can occur when the two parents have complementary genetic factors. Each parent may possess different beneficial alleles or genetic variations that, when combined in the offspring, result in a more complete set of functional genes. This complementary effect can lead to improved overall performance and adaptive traits. 4. Gene interaction and epistasis. Heterosis can arise from the interaction between different genes or epistasis, where the effect of one gene is dependent on the presence of another gene. The combination of different alleles at interacting gene loci can create a favorable genetic background that enhances certain traits or physiological processes. 5. Increased genetic diversity. Heterosis is often associated with increased genetic diversity. Hybrid offspring inherit a greater combination of genetic variations from both parents, which can provide a broader range of adaptability, resilience, and robustness to environmental stresses. This increased genetic diversity allows for more efficient resource utilization and improved overall fitness. At the molecular level, the basis of heterosis involves complex mechanisms and interactions among genes, regulatory elements, and molecular pathways. Several molecular processes have been proposed to contribute to heterosis, including times differential gene expression. Hybrids often exhibit altered patterns of gene expression compared to their parents. This differential gene expression can result from the interaction and combination of regulatory elements from both parents, leading to enhanced or novel gene expression profiles. Times transgressive gene expression. Heterosis can involve the activation or suppression of genes that are not expressed in the parents. Transgressive gene expression allows for the combination of favorable traits from both parents and the expression of novel phenotypes in the hybrid offspring. Times increased metabolic efficiency. Hybrids may exhibit improved metabolic efficiency, allowing for more efficient nutrient uptake, utilization, and energy production. This improved metabolic efficiency can contribute to enhanced growth, yield, and overall performance. Times regulatory networks and signaling pathways. The combination of parental genetic material in hybrids can lead to the rewiring or modification of regulatory networks and signaling pathways. This rewiring can result in altered responses to environmental cues, improved stress tolerance, and enhanced physiological processes. It's important to note that heterosis is a complex phenomenon influenced by multiple genetic and molecular factors. The specific mechanisms and contributions may vary depending on the species, traits, and genetic backgrounds involved. Researchers continue to investigate and unravel the molecular basis of heterosis to better understand and harness its potential for crop improvement and animal breeding. Classify types of male sterility and self-incompatibility system in plants. Describe the limitations of cytoplasmic genetic male sterility system in hybrid seed production. Types of male sterility. 1. Genetic male sterility. GMS. Genetic male sterility is caused by genetic factors that disrupt the development or function of male reproductive organs, resulting in the inability to produce viable pollen. It is inherited and can be controlled through breeding programs. 2. Cytoplasmic male sterility. CMS. Cytoplasmic male sterility is caused by the interaction between the nuclear and cytoplasmic genomes. The male sterility trait is controlled by the cytoplasmic, maternally inherited, genes, while the fertility-restoring genes are present in the nuclear genome. This system is widely used in hybrid seed production. 3. Environmental male sterility. EMS. Environmental male sterility occurs due to environmental factors such as temperature, light, or hormonal imbalances that disrupt pollen development or function. It is reversible and can vary based on the environmental conditions. Self-incompatibility systems. 1. Gametophytic self-incompatibility, GSI. In GSI, the determination of self-incompatibility is controlled by the genotype of the pollen grain, male gametophyte. The recognition and rejection of self-pollen occur within the female reproductive organ based on the interaction between specific alleles present in the pollen and the pistil. 2. Sporophytic self-incompatibility, SSI. In SSI, the recognition and rejection of self-pollen are controlled by the genotype of the sporophyte, the plant producing the pollen. The genotype of the pistil does not play a role in self-incompatibility determination. Limitations of cytoplasmic genetic male sterility, CGMS, in hybrid seed production. While cytoplasmic genetic male sterility, CGMS, has been widely utilized in hybrid seed production, it does have some limitations, including 1. Dependent on maintainer lines. CGMS requires the presence of a separate, maintainer, line to ensure the continuous production of male sterile plants. These maintainer lines need to be maintained and propagated alongside the male sterile lines, adding complexity and cost to the hybrid seed production process. 2. Risk of cytoplasmic contamination. There is a risk of cytoplasmic contamination in hybrid seed production when pollen from the maintainer line or other non-desirable pollen sources enters the female reproductive organs of the male sterile line. This can result in the production of partially or completely fertile seeds, reducing the purity and quality of the hybrid seeds. 3. Difficulty in developing new male sterile lines. Developing new male sterile lines with different desirable traits can be challenging. It often requires the transfer of cytoplasmic genes through backcrossing or other breeding techniques, which can be time-consuming and labor-intensive. 4. Limited genetic variation. The use of a specific cytoplasmic male sterile line restricts the genetic variation available for hybrid seed production. The male sterile line may lack certain desirable traits, and the limited genetic diversity may affect the overall adaptability and performance of the hybrid offspring. 5. Potential for resurgence of fertility. In some cases, there may be instances of fertility restoration in the male sterile lines due to the evolution of suppressor genes or spontaneous mutations. This can result in the production of self-pollinated or partially fertile plants, reducing the efficiency and reliability of hybrid seed production. To overcome these limitations, alternative approaches such as the use of genetically modified male sterility systems or other breeding techniques are being explored to enhance hybrid seed production and address the challenges associated with cytoplasmic genetic male sterility. What is clone? What do you understand by clonal selection? Describe the various steps involved in clonal selection. A clone refers to an exact genetic replica or copy of an organism, cell, or DNA fragment. Cloning can occur naturally through asexual reproduction, where offspring are derived from a single parent without genetic recombination. It can also be achieved artificially in the laboratory through techniques such as somatic cell nuclear transfer or cloning vectors. Clonal selection, in the context of plant breeding, refers to the process of selecting and propagating a superior individual plant or group of plants that possess desired traits. The selected plants are genetically identical to the parent plant, making them clones. Clonal selection allows for the preservation and multiplication of specific genetic traits and is commonly used in horticulture, forestry, and certain crop production systems. The steps involved in clonal selection are as follows. 1. Phenotypic evaluation. The initial step in clonal selection is the identification and evaluation of individuals with desirable traits. This can involve assessing various characteristics such as yield, disease resistance, growth habit, flower color, or any other traits of interest. 2. Propagation. Once a plant with desirable traits is identified, it is propagated asexually to produce multiple clones. This can be done through various methods such as stem cuttings, tissue culture, layering, or grafting, depending on the plant species and propagation requirements. 3. Establishment of clonal lines. The selected clones are grown and established as separate clonal lines. These clonal lines are carefully maintained and monitored to ensure the preservation of their genetic identity and desired traits. 4. Field evaluation. The established clonal lines are then evaluated under field conditions to assess their performance and stability. This step helps confirm the consistency of the desired traits across different environments and seasons. 5. Selection and multiplication. After field evaluation, further selection is carried out to identify the best performing clonal lines based on the desired traits. These selected lines are then multiplied through vegetative propagation methods to produce larger quantities of genetically identical plants. 6. Commercialization and distribution. The successfully selected and multiplied clonal lines are commercially propagated and made available to farmers, nurseries, or other end users. This ensures the widespread distribution and availability of plants with the desired traits. Clonal selection allows for the rapid multiplication of plants with known characteristics and desirable traits. It is advantageous in situations where sexual reproduction may result in the loss of desired traits due to genetic recombination. However, it is important to periodically introduce genetic diversity through sexual reproduction or other means to avoid the potential drawbacks associated with limited genetic variation in clonal populations, such as vulnerability to diseases or environmental changes. What do you understand by center of origin? Discuss the law of homologous series of variation. Center of origin refers to the geographic region or area where a particular species or group of related species is believed to have originated and diversified naturally over evolutionary time. It is the place where the wild ancestors of domesticated plants or animals are presumed to have originated and from where they dispersed or were later cultivated. The concept of the center of origin is based on the observation that certain regions possess a high diversity of related species, along with a wide range of genetic variation within those species. These regions are often characterized by favorable environmental conditions, such as suitable climate, soil types, and ecological niches that allowed for the evolution and adaptation of various species. The concept of the center of origin is important in agriculture and biodiversity conservation as it provides insights into the genetic diversity and potential of different species. Understanding the center of origin helps in identifying valuable genetic resources and conservation priorities, as well as guiding breeding and conservation efforts to ensure the preservation of genetic diversity. Law of homologous series of variation. The law of homologous series of variation, also known as the law of homologous characters, was proposed by Russian botanist Nikolai Vavilov. It states that related species or varieties within a plant or animal group tend to exhibit similar patterns of variation and possess common traits or characteristics. According to this law, species within a particular taxonomic group share a common genetic and evolutionary background. They have similar genetic structures and tend to show variation in a systematic manner. This means that when studying a group of related species or varieties, one can observe a series of variations that are similar but with progressive changes or modifications. For example, within a plant genus, different species may show variations in leaf shape, flower color, or fruit size, but these variations follow a pattern that is characteristic of that genus. The law of homologous series of variation highlights the presence of a common genetic basis for the variations observed within a taxonomic group. This law has important implications in plant breeding and taxonomy. It helps breeders understand the range of variation that can be expected within a species or group of related species, allowing them to select and develop new varieties with desired traits. It also aids taxonomists in classifying and organizing species based on shared characteristics and evolutionary relationships. Overall, the law of homologous series of variation provides a framework for understanding the patterns of variation within related species or varieties and offers insights into the genetic and evolutionary relationships within a taxonomic group. Describe self-pollination, cross-pollination and often cross-pollination. Discuss different mechanisms for facilitating self-pollination and cross-pollination. Self-pollination, cross-pollination, and often cross-pollination are different methods by which plants achieve pollination, the transfer of pollen from the male reproductive organ, anther, to the female reproductive organ, stigma, of a flower. These methods can vary based on the plant species and their reproductive strategies. 1. Self-pollination. Self-pollination occurs when pollen from the anther of a flower is transferred to the stigma of the same flower or another flower on the same plant. In self-pollinating plants, the pollen can reach the stigma through various mechanisms, including times callistogamy. Some plants have specialized flowers called callistogamous flowers that remain closed, preventing the entry of external pollinators. These flowers self-pollinate inside the closed structure. Times homogamy. Homogamous flowers have both male and female reproductive structures, anthers and stigma, positioned closely together, facilitating self-pollination. Times autogamy. Autogamy refers to the self-fertilization of a flower where pollen is transferred to the stigma within the same flower. This can occur due to the arrangement of the anther and stigma or through various self-pollination mechanisms. 2. Cross-pollination. Cross-pollination involves the transfer of pollen from the anther of a flower to the stigma of a different flower on a separate plant. This method promotes genetic diversity and often relies on external agents or mechanisms to transfer the pollen, such as times wind pollination. Anemophily vine-pollinated plants produce large quantities of lightweight and easily dispersed pollen. The pollen is released into the air and carried by the wind to neighboring flowers for pollination. Times insect pollination. Entomophily. Many plants rely on insects, such as bees, butterflies, or beetles, to transfer pollen between flowers. These plants often have showy flowers with attractive colors, fragrances, and nectar to attract pollinators. Times animal pollination. Zoophily. Animals like birds, bats, or small mammals can act as pollinators. They are attracted to flowers with specific adaptations, such as bright colors, long tubular structures, or copious nectar production, to facilitate pollen transfer. 3. Often cross-pollination. Some plants have the ability to self-pollinate but have mechanisms in place to promote cross-pollination, which leads to increased genetic diversity. These mechanisms include times dichogamy. Dichogamous flowers have male and female reproductive structures that mature at different times. This temporal separation prevents self-pollination and encourages cross-pollination by ensuring the pollen is transferred from a different flower. Times heterostyly. Heterostylous plants possess flowers with different lengths of stigma and stamen. This arrangement promotes cross-pollination as the length of the reproductive structures encourages pollen transfer between different plants. Times hypogamy. Hypogamous flowers have structural barriers, such as the position of the stigma and anthers at different heights or with physical barriers, that prevent self-pollination and encourage cross-pollination. These mechanisms and strategies have evolved in plants to optimize reproductive success and genetic diversity. By employing self-pollination, cross-pollination, or often cross-pollination, plants can adapt to their specific ecological niche and ensure the continuity of their species. What is the role and importance of seed treatment campaign in India in the context of quality crop establishment? In India, seed treatment campaigns play a crucial role in promoting quality crop establishment and improving agricultural productivity. Seed treatment involves the application of chemicals, biological agents, or other treatments to seeds before sowing. The goal is to protect the seeds from pests, diseases, and environmental stresses, as well as to enhance germination and seedling vigor. Here is the role and importance of seed treatment campaigns in India. 1. Disease and pest management. Seed treatment helps in controlling seed-borne diseases and pests that can negatively impact crop establishment and yield. Fungal, bacterial, and viral diseases can be effectively managed by treating seeds with fungicides, bactericides, or insecticides. This reduces the risk of disease transmission from seeds to young seedlings and promotes healthier plant growth. 2. Increased germination and vigor. Seed treatments can improve seed germination rates and seedling vigor, leading to better crop establishment. Treatments like priming, coating, or nutrient enhancement help overcome dormancy, stimulate early root and shoot development, and provide essential nutrients to the emerging seedlings. This enhances crop uniformity and early plant growth. 3. Protection against abiotic stresses. Seeds are exposed to various abiotic stresses, such as extreme temperatures, drought, and salinity, during germination and early growth stages. Seed treatments with stress-tolerant agents or biostimulants can help seeds withstand adverse environmental conditions, enhance stress tolerance, and improve overall crop performance. 4. Reduced dependence on chemicals. Seed treatment campaigns promote the use of environmentally friendly and safer alternatives to chemical pesticides. Biological agents, biopesticides, or biocontrol agents are often used in seed treatments, reducing the reliance on synthetic chemicals and minimizing their potential negative impacts on the environment and human health. 5. Adoption of good agricultural practices. Seed treatment campaigns provide an opportunity to educate farmers about the importance of adopting good agricultural practices, including proper seed selection, treatment, and handling. Training programs and awareness campaigns are conducted to promote the use of quality seeds, proper storage, and application techniques. 6. Enhanced productivity and yield. Quality crop establishment is vital for achieving higher agricultural productivity and crop yields. By ensuring healthy seedlings, improved germination, and early plant vigor, seed treatment campaigns contribute to the overall success of crop production. This leads to higher yields, improved crop quality, and increased farmer income. The Government of India, agricultural institutions, and seed companies actively promote seed treatment campaigns through various initiatives, including awareness programs, farmer training, subsidized seed treatment services, and the availability of quality seed treatment products. These efforts help farmers adopt seed treatment practices and contribute to sustainable agriculture, crop health, and food security in the country. What is DNA fingerprinting? Discuss its applied aspects in modern agriculture. DNA fingerprinting, also known as DNA profiling or genetic fingerprinting, is a technique used to identify and analyze the unique patterns of DNA sequences within an individual's genome. It involves the extraction, amplification, and analysis of specific DNA regions, such as microsatellites or short tandem repeats, STRs, that exhibit high variability among individuals. In modern agriculture, DNA fingerprinting has several applied aspects that contribute to various areas of crop improvement and management. 1. Genetic identification and verification. DNA fingerprinting is used to identify and verify the genetic identity of plants and agricultural crops. It helps distinguish different varieties or cultivars, ensuring the authenticity and purity of seeds or plant material. This is particularly important for intellectual property rights, certification, and protection against fraud or mislabeling. 2. Parentage determination. DNA fingerprinting can be employed to determine the parentage of crops, especially in hybrid breeding programs. By analyzing the DNA profiles of potential parents and offspring, breeders can identify the true parentage and confirm the success of hybridization, ensuring the desired traits are passed on to subsequent generations. 3. Marker-assisted selection. MAS DNA fingerprinting allows for the identification and selection of plants or varieties carrying specific genes or traits of interest. Molecular markers linked to important agronomic traits can be used to screen and select plants at early stages, reducing the time and resources required for traditional phenotypic selection. This accelerates the breeding process and enables the development of improved crop varieties with desired traits. 4. Disease resistance screening. DNA fingerprinting is used to identify genetic markers associated with disease resistance or susceptibility in crops. By screening plant DNA, breeders can identify individuals or varieties that carry resistance genes against specific pathogens or pests. This information guides breeding efforts to develop resistant crop varieties and implement effective disease management strategies. 5. Genetic diversity assessment. DNA fingerprinting provides insights into the genetic diversity and population structure of crops. By analyzing DNA profiles of different individuals or varieties, scientists can assess the genetic variation, relatedness, and population dynamics within a crop species. This information is valuable for germplasm conservation, crop improvement, and the development of breeding strategies to enhance genetic diversity. 6. Traceability and quality control. DNA fingerprinting is used to establish traceability and quality control in agricultural supply chains. By analyzing the DNA profiles of crops at different stages, from seed to final product, it ensures product authenticity, facilitates traceability, and aids in detecting adulteration or contamination in the supply chain. Overall, DNA fingerprinting plays a significant role in modern agriculture by providing valuable tools for genetic identification, parentage determination, marker-assisted selection, disease resistance screening, genetic diversity assessment, and traceability. These applications contribute to crop improvement, quality assurance, and sustainable agricultural practices. Describe the mechanisms involved in absorption of water in plants. Explain the factors affecting the water absorption rate in plants. The absorption of water in plants occurs through the roots and involves various mechanisms and processes. Here are the key mechanisms involved in water absorption. 1. Root hair system. The root hairs, which are fine projections on the surface of root epidermal cells, greatly increase the surface area of the root for water absorption. The root hairs create a zone called the root hair zone, which is the primary site for water uptake. 2. Osmosis. Water moves into the plant roots through the process of osmosis. Osmosis occurs due to the difference in water potential between the soil solution and the root cells. Water moves from an area of higher water potential, the soil, to an area of lower water potential, the root cells, through osmotic pressure. 3. Root pressure. In some plant species, root pressure can contribute to water absorption. Root pressure is generated by active transport of ions into the root cells, which creates a pressure potential. This pressure pushes water up the plant, aiding in its movement through the xylem vessels. 4. Capillary action. Capillary action refers to the ability of water to move upwards in narrow spaces against gravity. It occurs due to the cohesive and adhesive properties of water and the presence of tiny pores and narrow spaces in the soil and root tissues. Capillary action allows water to be drawn upwards in the root system. Factors affecting the water absorption rate in plants include 1. Soil moisture. The availability of water in the soil is a crucial factor affecting water absorption. When the soil is dry or waterlogged, it can limit the rate of water uptake by the roots. Optimal soil moisture conditions promote efficient water absorption. 2. Root architecture and surface area. The presence of a well-developed root system with extensive branching and a large surface area enhances water absorption. The density and length of root hairs also influence the rate of water uptake. 3. Transpiration pull. Transpiration, the loss of water vapor through the leaves, creates a negative pressure or tension in the xylem vessels, resulting in a transpiration pull. This pull helps in the upward movement of water from the roots to the above ground parts of the plant. Factors that affect transpiration, such as temperature, humidity, and air movement, indirectly influence water absorption. 4. Soil texture and structure. The texture and structure of the soil can affect water absorption. Sandy soils drain quickly but may have lower water holding capacity, while clay soils hold water for longer but can become compacted, affecting root penetration and water uptake. 5. Root environment. The presence of root pathogens or excessive salt concentrations in the soil can negatively impact water absorption. Pathogens can cause damage to the root system, reducing the ability to absorb water. High salt concentrations can create an osmotic imbalance, hindering water uptake. 6. Plant physiology. The physiological state of the plant, including the stomatal regulation of transpiration and the plant's water status, can influence the rate of water absorption. Factors such as plant age, hormonal signals, and internal water potential affect water uptake. Understanding the mechanisms of water absorption and the factors that influence it is important for optimizing irrigation practices, managing water stress, and promoting healthy plant growth and development. Define seed viability. Describe in brief the metabolic aspects of seed germination. Seed viability refers to the ability of a seed to germinate and develop into a viable and healthy plant under suitable conditions. It represents the proportion of seeds in a population that are capable of germination. A viable seed possesses the necessary internal structures, biochemical processes, and genetic information to initiate and sustain germination. The metabolic aspects of seed germination involve a series of complex biochemical changes within the seed, triggered by environmental cues such as moisture, temperature, and light. These metabolic processes are crucial for breaking dormancy and activating growth. Here are the key stages involved. 1. Imbibition. When a viable seed encounters suitable conditions, it absorbs water through the seed coat in a process called imbibition. The seed swells, activating cellular activities and rehydrating the dormant embryo. 2. Respiration. With imbibition, respiration in the seed increases. Oxygen is utilized to break down stored carbohydrates, lipids, and proteins, energy required for further growth. 3. Activation of enzymes. The increase in respiration triggers the activation of enzymes within the seed. These enzymes catalyze biochemical reactions, including the conversion of stored starches into simple sugars, which provide energy for growth. 4. Mobilization of reserves. The stored reserves, such as starches, oils, and proteins, are broken down into simpler molecules that can be transported to the growing embryo. These nutrients serve as a source of energy and building blocks for the developing plant. 5. Cell division and elongation. As the seed germinates, cells within the embryo start to divide and elongate, forming the radicle, embryonic root, and the plumule, embryonic shoot. The radicle emerges first, anchoring the seedling into the soil, while the plumule grows upward, seeking light. 6. Synthesis of new molecules. As growth continues, the seedlings synthesize new molecules, such as nucleic acids, proteins, and chlorophyll, which are necessary for sustaining growth and photosynthesis. 7. Emergence and establishment. Finally, the seedling emerges from the soil and begins to establish itself as an independent plant, capable of photosynthesis and further development. Overall, seed germination involves a coordinated series of metabolic processes that transform the dormant seed into an actively growing seedling. These processes require the utilization of stored reserves and the synthesis of new molecules to support the initial stages of plant growth. Define pests and pesticides in relation to pest management. Give a brief account of types and need for pesticide formulations. In the context of pest management, pests refer to organisms that interfere with human activities, cause damage to crops, livestock, structures, or transmit diseases. Pests can include insects, weeds, rodents, fungi, bacteria, and other organisms that pose a threat to human health, agriculture, or the environment. Pesticides are chemical or biological substances used to control or eliminate pests. They are designed to target specific pests and disrupt their life cycles, reduce their populations, or prevent damage to crops, structures, or public health. Pesticides can be classified into different categories based on their target pests and their mode of action. 1. Insecticides. These pesticides specifically target insects and are used to control or eradicate insect populations that damage crops, transmit diseases, or cause nuisance. Insecticides can be further classified into contact insecticides, kill insects upon contact, systemic insecticides, absorbed by plants and ingested by insects, and stomach poisons, ingested by insects and disrupt their digestive system. 2. Herbicides. Herbicides are used to control or eliminate weeds that compete with crops for nutrients, water, and sunlight. They are designed to selectively or non-selectively target plants and inhibit their growth or kill them. 3. Fungicides. Fungicides are used to control or prevent fungal diseases that affect plants, crops, or structures. They inhibit fungal growth, disrupt their reproductive cycles, or protect plants from fungal infections. 4. Rodenticides. These pesticides are used to control rodent populations, such as rats and mice, which can cause damage to crops, stored goods, or spread diseases. Rodenticides are designed to attract and kill rodents. 5. Bactericides and virucides. These pesticides target bacteria and viruses, respectively, and are used to control or prevent the spread of bacterial or viral diseases in plants, animals, or humans. The need for pesticide formulations arises from the desire to optimize their effectiveness, ease of application, and safety. Pesticide formulations are developed by combining active ingredients, the pesticide compounds, with other additives and carriers to enhance their stability, dispersibility, adhesion, and target specificity. Common types of pesticide formulations include 1. Liquid formulations. These include emulsifiable concentrates, solutions, suspensions, and aerosols. They are easy to apply, can cover large areas, and provide uniform distribution of the active ingredient. 2. Solid formulations. Solid formulations include dusts, granules, and tablets. They are often used for specific target areas or pests and can be applied by spreading or sprinkling. 3. Gas formulations. These are pesticides in the form of fumigants, which release toxic gases to control pests in enclosed spaces or stored commodities. Pesticide formulations are developed to ensure proper delivery, maximize efficacy, minimize environmental impact, and improve safety for applicators and the surrounding ecosystem. They undergo rigorous testing and regulation to ensure their effectiveness and adherence to safety standards. It is important to follow proper guidelines and regulations for the use of pesticides to mitigate potential risks and ensure responsible pest management practices. Narrate the role of post-harvest technology in minimizing losses in fruits and vegetables. Post-harvest technology plays a crucial role in minimizing losses in fruits and vegetables by implementing various practices and techniques that preserve the quality and extend the shelf life of these perishable commodities. Here is a narrative on the role of post-harvest technology in reducing losses. Once fruits and vegetables are harvested, they undergo physiological changes and become susceptible to various factors that can lead to spoilage and deterioration. Post-harvest losses occur due to factors such as physiological changes, microbial growth, mechanical damage, temperature fluctuations, and improper handling and storage practices. However, through the application of post-harvest technology, these losses can be significantly reduced, ensuring that a greater proportion of the harvested produce reaches consumers in a fresh and marketable condition. One of the key aspects of post-harvest technology is proper handling and transportation. It involves careful sorting, grading, and packing of fruits and vegetables to minimize physical damage. By employing gentle handling techniques and appropriate packaging materials, such as crates, cartons, or modified atmosphere packaging, the risk of bruising, abrasions, and other mechanical injuries can be minimized. Temperature management is another vital component of post-harvest technology. Most fruits and vegetables are highly perishable and are prone to rapid deterioration at ambient temperatures. Therefore, employing cooling techniques like refrigeration or controlled atmosphere storage can significantly extend the shelf life and maintain the quality of the produce. Cold storage facilities, such as walk-in coolers or cold rooms, help slow down physiological processes, reduce microbial growth, and prevent moisture loss, thereby preserving the texture, flavor, and nutritional content of the fruits and vegetables. Post-harvest treatments such as washing, sanitizing, and surface coatings can help reduce microbial contamination and extend the shelf life. These treatments, along with proper drying, can minimize the growth of spoilage-causing microorganisms and enhance the visual appeal of the produce. Additionally, post-harvest technology involves implementing quality control measures such as regular monitoring, inspection, and grading of the produce. By identifying and removing damaged or diseased fruits and vegetables, the risk of further spoilage can be mitigated, and only high-quality produce can be sent to the market. Furthermore, value addition techniques are employed in post-harvest technology to enhance the market value and utilization of fruits and vegetables. Processes like canning, freezing, drying, juicing, and making preserves or purees enable the preservation of seasonal produce for an extended period, thereby reducing losses and allowing consumers to enjoy them throughout the year. By implementing these post-harvest technologies and practices, the losses of fruits and vegetables can be minimized, leading to increased availability of nutritious produce, reduced wastage, and improved income for farmers and other stakeholders in the supply chain. The application of post-harvest technology not only ensures that consumers have access to fresh and safe produce but also contributes to food security, sustainable agriculture, and the reduction of food waste. What is plant quarantine? Describe the plant quarantine measures with suitable examples in Indian context. Plant quarantine refers to the set of measures and regulations implemented to prevent the introduction and spread of pests, diseases, and invasive plant species across international or regional borders. It involves the inspection, testing, and control of plants, plant products, and related materials to ensure their safe movement without posing a risk to agricultural, horticultural, or environmental systems. In the Indian context, several plant quarantine measures are in place to safeguard the country's plant health and agricultural resources. Here are some examples of plant quarantine measures implemented in India. 1. Phytosanitary certification. India requires phytosanitary certificates for the import and export of plants, plant products, and related materials. These certificates are issued by the National Plant Protection Organization, NPPO, or authorized agencies after verifying compliance with the phytosanitary requirements of the importing or exporting countries. The certification ensures that the consignments are free from regulated pests and diseases. 2. Pest risk analysis. PRA is conducted to assess the risks associated with the introduction or spread of pests through international trade. The Indian NPPO evaluates the potential risks posed by pests and determines appropriate phytosanitary measures to mitigate those risks. PRA helps in developing pest-specific regulations and guidelines for quarantine treatments, inspections, and other control measures. 3. Quarantine inspections. At ports of entry, quarantine inspections are conducted on imported plant material to check for the presence of pests, diseases, or other regulated organisms. Inspections may involve visual examination, laboratory testing, or other diagnostic techniques to identify and intercept potential threats. Quarantine officers have the authority to reject or subject consignments to treatment or disinfection if pests or diseases are detected. 4. Quarantine treatments. Some plant material, especially fruits, vegetables, and cut flowers, may undergo quarantine treatments to ensure the elimination or suppression of pests and pathogens. For example, hot water treatment or vapor heat treatment may be applied to perishable commodities to control fruit flies and other insects. Fumigation with approved chemicals or irradiation may be used to disinfect stored products or grains. 5. Border surveillance. India maintains a system of border surveillance to monitor and detect the entry of exotic pests or diseases. Inspection stations, quarantine checkposts, and surveillance networks are established to track the movement of plant material and implement appropriate control measures. In case of an outbreak or detection of a new pest, immediate actions are taken to contain and eradicate the pest. 6. Pest eradication programs. If an invasive pest or disease is detected within the country, quarantine authorities may initiate eradication programs to prevent its spread. These programs involve the use of chemical treatments, biological control agents, or other measures to eliminate or manage the pests effectively. The implementation of plant quarantine measures in India is essential for safeguarding agricultural productivity, protecting native plant species, and ensuring food security. These measures help prevent the introduction and establishment of invasive pests, diseases, and weeds, which can have detrimental effects on crop production, natural ecosystems, and the economy. By regulating the movement of plant material, India aims to maintain plant health and minimize the risks associated with international trade in plants and plant products. Discuss the importance of greenhouse in agriculture. Greenhouses play a significant role in modern agriculture and offer numerous benefits that contribute to the productivity, quality, and sustainability of crop production. Here are some key reasons why greenhouses are important in agriculture. 1. Extended growing season. Greenhouses provide a controlled environment that allows farmers to extend the growing season beyond the natural limitations of the climate. By creating a favorable microclimate, greenhouses enable the cultivation of crops throughout the year, regardless of seasonal changes, extreme temperatures, or adverse weather conditions. This ensures a consistent and continuous supply of fresh produce, reducing reliance on specific growing seasons. 2. Enhanced crop protection. Greenhouses act as physical barriers that protect crops from external factors such as pests, diseases, and harsh weather conditions. The enclosed structure prevents the entry of insects, birds, and other pests, reducing the need for chemical pesticides. Moreover, greenhouses provide a shield against extreme temperatures, frost, heavy rain, hailstorms, and strong winds, minimizing crop damage and losses. 3. Improved crop quality and yield. Greenhouse environments can be carefully optimized to provide ideal growing conditions for crops. Factors such as temperature, humidity, light intensity, and CO2 levels can be precisely controlled, creating an optimal environment that promotes healthy growth and development. This results in improved crop quality, enhanced flavors, increased nutritional value, and higher yields compared to open field cultivation. 4. Efficient resource utilization. Greenhouses offer opportunities for resource efficiency in agriculture. By controlling the inputs, such as water and fertilizers, within the greenhouse environment, farmers can minimize waste and optimize resource utilization. Irrigation systems can be tailored to meet specific crop needs, reducing water consumption. Nutrient delivery can be precisely managed, resulting in reduced fertilizer use and less environmental pollution. Greenhouses also enable efficient land use as they can be designed to maximize the production area by utilizing vertical growing techniques. 5. Year-round production of specialty crops. Greenhouses provide an ideal environment for growing specialty crops that may be challenging or impractical to cultivate in the local climate or soil conditions. This includes high-value crops like exotic fruits, vegetables, herbs, flowers, and medicinal plants. Greenhouses enable farmers to meet the increasing demand for these specialty crops, offering market opportunities and diversification of agricultural production. 6. Climate change resilience. Greenhouses offer a means to adapt to the effects of climate change. As global temperatures rise and weather patterns become more unpredictable, greenhouses provide a controlled environment that buffers crops from extreme conditions. They can help mitigate the impacts of heat waves, droughts, heavy rainfall, and erratic temperature fluctuations, ensuring crop stability and food security. Overall, greenhouses provide a protected and optimized environment for crop cultivation, allowing for year-round production, improved crop quality, enhanced resource efficiency, and climate resilience. They offer a sustainable approach to agriculture, reducing the reliance on pesticides, conserving resources, and contributing to food production in regions with challenging climates. What are enzymes? Describe key log theory with respect to mode of enzyme action. Enzymes are biological molecules, specifically proteins, that act as catalysts in various biochemical reactions. They accelerate the rate of chemical reactions by lowering the activation energy required for the reaction to occur. Enzymes are highly specific, each one designed to catalyze a particular reaction or a group of related reactions. The key log theory, also known as the lock and key model, is a concept that explains the mode of enzyme action and the specificity of enzyme-substrate interactions. According to this theory, one enzyme, log. The enzyme is represented as the log in the analogy. It has a specific three-dimensional structure with an active site, a region where the substrate molecules can bind. 2. Substrate, key. The substrate is represented as the key in the analogy. It is the molecule upon which the enzyme acts. The substrate has a complementary shape to the active site of the enzyme. 3. Enzyme-substrate interaction. The key log theory proposes that the enzyme's active site and the substrate have complementary shapes, much like a lock and a key. Only when the substrate's shape precisely matches the active site's shape can the substrate bind to the enzyme. 4. Enzyme-substrate complex. When the substrate binds to the active site, an enzyme-substrate complex is formed. The complex is held together by weak chemical bonds, such as hydrogen bonds, electrostatic interactions, or van der Waals forces. 5. Enzyme-substrate reaction. Once the substrate is bound to the enzyme catalyzes the conversion of the substrate into the product, S, of the reaction. The enzyme facilitates this transformation by providing an environment that stabilizes the transition state of the reaction, reducing the activation energy required for the reaction to occur. 6. Product release. After the reaction is complete, the product, S, of the reaction dissociate from the active site, leaving the enzyme free to bind with other substrate molecules and repeat the catalytic process. The key log theory emphasizes the importance of the specific structural complementarity between the enzyme and substrate for efficient catalysis. The shape, charge distribution, and functional groups of the substrate must align precisely with the active site's structure and chemical properties of the enzyme. However, it's important to note that the key log theory represents a simplified model, and the actual mechanism of enzyme action can be more dynamic and flexible. In some cases, enzymes undergo conformational changes upon substrate binding, allowing for a more precise fit between the enzyme and substrate. Overall, the key log theory provides a conceptual framework to understand how enzymes selectively bind to specific substrates and facilitate chemical reactions, showcasing the remarkable specificity and efficiency of enzymatic catalysis. What is plant physiological stress? Describe morphological and physiochemical changes taking place in plants under drought condition. Plant physiological stress refers to the adverse conditions or factors that disrupt the normal physiological functions of plants, impair their growth, and decrease their overall productivity. One of the most common and severe forms of physiological stress for plants is drought, which occurs when plants experience an insufficient supply of water to meet their physiological needs. Under drought conditions, plants undergo various morphological and physiochemical changes as adaptive responses to minimize water loss and maintain their survival. Here is a description of some of the key changes that take place in plants under drought conditions. Morphological changes. 1. Leaf rolling or curling. In response to water scarcity, plants may exhibit leaf rolling or curling, which reduces the leaf surface area exposed to the air, minimizing water loss through transpiration. 2. Reduced leaf size. Drought-stressed plants often exhibit smaller leaf size as a result of reduced cell expansion and growth. This helps to limit water loss through the stomata. 3. Stunted growth. Drought stress inhibits cell division and elongation, leading to overall reduced growth and development of the plant, resulting in stunted stature. 4. Increased root growth. Plants respond to drought stress by increasing root growth to explore a larger soil volume in search of available water. This is an adaptive mechanism to enhance water uptake from deeper soil layers. Physiochemical changes. 1. Stomatal closure. One of the primary responses to drought stress is the closure of stomata, small openings on the leaf surface, to reduce water loss through transpiration. Stomatal closure limits gas exchange, including the uptake of carbon dioxide for photosynthesis, which can negatively impact plant growth. 2. Accumulation of compatible solutes. Drought-stressed plants often accumulate compatible solutes, such as sugars, amino acids, and organic acids, which act as osmoprotectants. These solutes help maintain cellular water potential and stabilize protein structures, protecting the plant cells from dehydration damage. 3. Increased production of stress-related hormones. Drought stress triggers the synthesis and release of stress-related hormones, such as abscisic acid, ABA. ABA plays a crucial role in regulating stomatal closure, inhibiting growth, and promoting the synthesis of stress proteins that protect the plant from cellular damage caused by drought. 4. Enhanced antioxidant defense. Drought stress leads to the accumulation of reactive oxygen species, ROS, in plant cells, which can cause oxidative damage. To counteract this, plants activate their antioxidant defense systems, including the production of enzymes like superoxide dismutase, SOD, catalase, CAT, and peroxidase, POD, which scavenge ROS and protect plant tissues from oxidative stress. 5. Alteration in metabolic pathways. Drought stress can cause changes in metabolic pathways within plant cells. For example, there may be a shift from aerobic respiration to anaerobic respiration in some tissues to maintain energy production under low oxygen availability. These morphological and physiochemical changes in plants under drought conditions are adaptive responses aimed at reducing water loss, maintaining cellular water potential, and protecting plants from damage caused by water scarcity. These responses allow plants to survive and endure until water becomes available again, enabling their recovery and growth once favorable conditions are restored. What do you understand by vernalization? Describe its practical application in crops. Vernalization refers to the process by which plants require exposure to a prolonged period of cold temperatures to induce or promote flowering. It is a specific environmental stimulus that triggers the transition from the vegetative to the reproductive phase in certain plant species. Vernalization is particularly important for biennial and winter annual plants that require a period of cold exposure before they can flower. During vernalization, the exposure to cold temperatures brings about physiological and biochemical changes in the plant, which eventually enable flowering when favorable conditions return. The exact mechanisms of vernalization vary among different plant species, but the process generally involves epigenetic modifications, such as changes in gene expression and chromatin structure. The practical application of vernalization in crops is significant for several reasons. 1. Promoting flowering in winter and spring crops. Vernalization allows the cultivation of winter and spring crops that require a period of cold exposure to initiate flowering. For example, crops like winter wheat, barley, rye, and certain brassica vegetables, such as cabbage and kale, require vernalization to transition from the vegetative stage to flowering and subsequent seed or fruit production. 2. Regulation of flowering time. Vernalization helps control and synchronize the flowering time of certain crops, ensuring that they flower at the desired time to optimize yield and quality. This is particularly important in the case of biennial crops, where vernalization in the first year induces flowering in the subsequent year. 3. Adaptation to specific climates. Vernalization allows plants to adapt and thrive in regions with cold winters or seasonal variations. By requiring exposure to cold temperatures, vernalization ensures that plants flower during suitable growing seasons, avoiding premature flowering and potential damage from freezing temperatures. 4. Crop breeding and improvement. Understanding the vernalization requirements of crops is crucial for crop breeding programs. Breeders can select and develop varieties that have specific vernalization requirements, allowing them to adapt crops to different climatic conditions and optimize their performance in specific environments. 5. Manipulation of flowering time. The knowledge of vernalization mechanisms can be applied in crop management strategies to manipulate or control flowering time. This can be useful for extending or shortening the growing season, adjusting planting schedules, and optimizing crop production in different regions or climates. Overall, vernalization is an important physiological process in certain plant species that allows for proper flowering and reproductive development. Understanding and harnessing the principles of vernalization have practical implications in crop production, including the cultivation of winter and spring crops, regulation of flowering time, adaptation to specific climates, and crop breeding efforts for improved performance and productivity. What do you mean by economic injury level, EIL, and economic threshold level, ETL? Describe their roles in classifying the pests in relevance to integrated pest management, IPM. Economic injury level, EIL, and economic threshold level, ETL, are concepts used in integrated pest management, IPM, to determine the point at which pest populations or damage levels reach a level where intervention measures become economically justified. These concepts help guide decision-making regarding the application of pest control measures. The economic injury level, EIL, refers to the pest population density or level of damage at which the monetary losses caused by the pest exceed the cost of implementing control measures. It is the point where the economic impact of the pest becomes significant enough to warrant intervention. The EIL is determined by considering factors such as crop value, crop growth stage, pest biology, and the cost of control measures. The goal is to apply control measures before the pest population or damage reaches a level that would cause unacceptable economic losses. The economic threshold level, ETL, is the pest population density or damage level at which the application of control measures is deemed necessary to prevent the pest population from reaching the EIL. The ETL is typically set at a lower pest population or damage level compared to the EIL. By intervening at the ETL, pest populations can be suppressed and kept below the EIL, minimizing economic losses. In the context of integrated pest management, IPM, the concepts of EIL and ETL are essential for making informed decisions about pest control strategies. IPM aims to manage pests in a sustainable and economically viable manner, minimizing reliance on pesticides and considering ecological and economic factors. The roles of EIL and ETL in IPM include 1. Monitoring and scouting. Regular monitoring of pest populations and damage levels allows for the determination of ETL and EIL thresholds. Pest populations are assessed through field observations, trapping, sampling, and other monitoring techniques. Monitoring helps to detect pest outbreaks and enables timely intervention before economic losses occur. 2. Decision-making. EIL and ETL provide decision-makers, such as farmers or pest management professionals, with clear guidelines for determining when and how to implement control measures. Based on the pest population density or damage level in relation to the ETL, decisions can be made regarding the initiation and intensity of pest control actions. 3. Targeted pest control. IPM emphasizes the use of multiple pest management strategies and the integration of various control methods, such as cultural practices, biological control, and judicious use of pesticides. By implementing control measures at the ETL, the focus is on suppressing pest populations to a tolerable level while minimizing environmental impacts and reducing the economic costs associated with excessive control measures. 4. Cost effectiveness. EIL and ETL considerations help ensure that pest control measures are economically justified. By avoiding unnecessary control measures when pest populations are below the ETL, resources can be allocated efficiently, reducing unnecessary pesticide use and associated costs. In summary, the concepts of EIL and ETL are crucial components of IPM, providing thresholds for pest population densities or damage levels that trigger appropriate pest management actions. By utilizing EIL and ETL in IPM strategies, pest control efforts can be targeted, effective, and economically sound, promoting sustainable pest management practices. Describe the importance of fruit cultivation in India. Discuss the role of horticulture with respect to poverty alleviation and nutritional security. Fruit cultivation plays a vital role in India due to its economic, nutritional, and social significance. Here are some key points highlighting the importance of fruit cultivation in India. 1. Economic contribution. Fruit cultivation is a significant contributor to India's agricultural economy. It provides employment opportunities for millions of people, both in rural areas and along the entire value chain, including farming, harvesting, processing, distribution, and marketing. The fruit industry generates income for farmers, laborers, traders, and other stakeholders, contributing to rural development and economic growth. 2. Diversification of agriculture. Fruit cultivation offers a means of diversifying agricultural production. It allows farmers to grow a wide range of fruit crops, including mangoes, bananas, citrus fruits, apples, grapes, and many more. Diversification helps reduce the risks associated with monoculture, enhances farmers' resilience to market fluctuations and climate variability, and provides opportunities for crop rotation and intercropping. 3. Nutritional security. Fruits are a rich source of essential vitamins, minerals, fiber, and antioxidants, playing a crucial role in promoting a balanced and nutritious diet. Fruit consumption is associated with improved health outcomes, including the prevention of chronic diseases. The availability and accessibility of a diverse range of fruits contribute to enhancing nutritional security and addressing malnutrition issues in the country. 4. Export potential. India has a favorable climate and diverse agroecological zones, which allow for the production of a wide range of fruits. Many Indian fruits, such as mangoes, grapes, bananas, and pomegranates, have excellent export potential. Fruit exports contribute to foreign exchange earnings, boost the country's agricultural exports, and enhance its global reputation as a supplier of high-quality fruits. 5. Poverty alleviation and rural development. Fruit cultivation plays a crucial role in poverty alleviation and rural development. By providing income-generating opportunities, particularly for small and marginal farmers, fruit cultivation helps improve livelihoods, reduce poverty, and bridge the income gap. It empowers farmers, especially women and marginalized communities, by creating avenues for entrepreneurship, value addition, and market integration. 6. Environmental sustainability. Fruit cultivation, particularly when practiced using sustainable and eco-friendly methods, can contribute to environmental conservation. Fruits are often grown using less water compared to many other crops, and agroforestry systems incorporating fruit trees can help sequester carbon, enhance biodiversity, and improve soil health. Horticulture, which encompasses fruit cultivation along with vegetable, flower, and ornamental plant production, plays a pivotal role in poverty alleviation and nutritional security. 1. Income generation. Horticulture, including fruit cultivation, provides diverse income-generating opportunities for smallholder farmers, particularly those with limited land resources. It offers higher value crops and enables farmers to earn higher returns compared to traditional cereal crops. 2. Employment opportunities. Horticulture activities create employment opportunities, both on farm and off farm, throughout the value chain. It generates jobs in farming, nurseries, post-harvest handling, packaging, transportation, processing, and marketing, leading to income generation and reduced unemployment. 3. Nutritional diversity. Horticulture, with its focus on diverse crops, contributes to enhancing dietary diversity and improving nutritional outcomes. Fruits and vegetables produced through horticulture practices provide essential nutrients, addressing micronutrient deficiencies and promoting better health. 4. Women empowerment. Horticulture, including fruit cultivation, often involves active participation by women in various stages of production, post-harvest activities, and value addition. This empowerment of women helps uplift their socio-economic status and promotes gender equality. 5. Market opportunities. The increasing demand for fresh fruits and vegetables, both domestically and globally, presents market opportunities for horticulture farmers. Access to markets, value addition, and market linkages enable farmers to fetch better prices, improve income, and enhance their economic well-being. In summary, fruit cultivation within the broader context of horticulture plays a multifaceted role in India, encompassing economic development, poverty alleviation, nutritional security, and environmental sustainability. It offers income generation, employment opportunities, diversified nutrition, and market potential, contributing to the overall well-being of farmers, rural communities, and the nation as a whole. What is protected cultivation? What are its problems and prospects in comparison to open cultivation? Protected cultivation, also known as greenhouse cultivation or controlled environment agriculture, refers to the practice of growing crops within a controlled environment, typically within structures such as greenhouses or polytunnels. In protected cultivation, environmental conditions such as temperature, humidity, light intensity, and air circulation are manipulated to optimize plant growth and protect crops from adverse weather conditions, pests, and diseases. While protected cultivation offers numerous advantages, it also has its share of problems and prospects when compared to open cultivation. Here is a comparison of the two. Problems of protected cultivation. 1. High initial investment. Setting up a protected cultivation system, including the construction of greenhouses or polytunnels, installation of environmental control systems, and purchase of equipment, requires a significant initial investment. This can be a barrier for small-scale farmers or those with limited financial resources. 2. Operational costs. Maintaining the controlled environment in protected cultivation systems involves ongoing operational costs. These include energy costs for heating, cooling, and lighting, as well as expenses for irrigation, ventilation, and pest control. These costs can be higher compared to open cultivation where reliance on natural conditions is more prevalent. 3. Technical expertise. Successful implementation of protected cultivation techniques requires technical knowledge and skills in greenhouse management, crop-specific requirements, climate control, irrigation, and pest management. Farmers may need training and support to effectively manage the complexities of protected cultivation systems. 4. Risk of crop failure. In protected cultivation, crops are more dependent on artificial inputs and technology. If there are failures or malfunctions in the environmental control systems, such as power outages or equipment breakdowns, it can lead to adverse effects on crops. The risk of crop failure due to technical issues or operator error can be higher compared to open cultivation. Prospects of protected cultivation. 1. Extended growing season. Protected cultivation allows farmers to extend the growing season by creating a favorable microclimate within the structures. It enables the cultivation of crops during adverse weather conditions, such as cold winters or excessive rainfall, which would otherwise be challenging or impossible in open fields. This can lead to increased productivity and a more consistent supply of crops. 2. Pest and disease management. Protected cultivation provides a physical barrier that prevents pests and diseases from directly accessing crops. This reduces the reliance on chemical pesticides and allows for more targeted and environmentally friendly pest management strategies, such as biological control methods. It can result in improved crop health and reduced crop losses due to pests and diseases. 3. Improved yield and quality. By controlling environmental factors, such as temperature, humidity, and light intensity, protected cultivation enables farmers to optimize crop growth and yield. It also provides better control over crop quality attributes, such as size, color, taste, and nutritional content, leading to the production of high-value crops that meet market demands. 4. Water and resource efficiency. Protected cultivation systems offer the opportunity for efficient water use through controlled irrigation methods, such as drip irrigation or hydroponics. Water and nutrient recycling systems can be implemented, reducing water and fertilizer wastage. Additionally, protected cultivation can result in more efficient land use and higher crop density, maximizing the productivity of limited land resources. 5. Climate change adaptation. Protected cultivation can help farmers adapt to the challenges posed by climate change. It provides a controlled environment that buffers crops from extreme weather events, temperature fluctuations, and other climate-related risks. It offers a means of climate-proofing agriculture and ensuring more stable crop production in the face of changing climatic conditions. In summary, protected cultivation offers significant prospects for improving crop productivity, quality, and resilience. It provides a controlled environment that protects crops from adverse weather conditions, pests, and diseases. However, it also presents challenges related to initial investment, operational costs, technical expertise, and the risk of crop failure. The prospects of protected cultivation lie in its ability to extend the growing season, enhance pest and disease management, improve yield and quality, increase water and resource efficiency, and adapt to climate change. What is the food production and consumption pattern of India in the last 50 years? India's food production and consumption pattern has undergone significant changes over the past 50 years. Here are some key trends and shifts observed during this period. 1. Green Revolution and increased food production. The 1960s and 1970s witnessed the introduction of the Green Revolution in India, which led to a significant increase in food production. High-yielding varieties of crops, improved irrigation infrastructure, and the use of chemical fertilizers and pesticides played a crucial role in boosting agricultural productivity, particularly in wheat and rice production. 2. Shift in crop preferences. With the Green Revolution, there was a shift in crop preferences towards wheat and rice, which became staple food crops. The production of these cereals increased significantly, leading to self-sufficiency in food grains and reducing dependence on imports. 3. Diversification of agriculture. Over time, there has been a growing recognition of the need for crop diversification to ensure balanced nutrition and sustainability. The cultivation of cash crops, horticulture crops, fruits and vegetables, oilseeds, pulses, and spices has increased to meet changing consumer preferences and nutritional requirements. 4. Changing dietary patterns. With increasing urbanization, changing lifestyles, and rising incomes, dietary patterns in India have been shifting. There has been a rise in the consumption of processed and packaged foods, including snacks, convenience foods, and beverages. The demand for meat, dairy products, and seafood has also increased due to changing preferences. 5. Increased per capita food availability. Over the past 50 years, per capita food availability has improved significantly in India. Increased food production, government interventions, and public distribution systems have helped ensure a relatively steady supply of food, although challenges related to distribution and access remain. 6. Nutritional challenges. While food availability has improved, India continues to face nutritional challenges. Malnutrition, particularly undernutrition and micronutrient deficiencies, remains a significant concern, especially among marginalized populations and in rural areas. Efforts are being made to address these challenges through various programs and initiatives focused on improving food security and nutrition. 7. Increasing food waste. Alongside the increase in food production and consumption, there has been a rise in food waste. Post-harvest losses, inefficient supply chains, inadequate storage facilities, and consumer behavior contribute to significant food wastage, leading to economic losses and resource inefficiencies. 8. Focus on sustainable agriculture. In recent years, there has been a growing emphasis on sustainable agriculture practices. Efforts are being made to promote organic farming, reduce the use of chemical inputs, conserve water resources, and adopt climate-smart agricultural practices to ensure long-term food security and environmental sustainability. It's important to note that India's food production and consumption patterns are diverse and vary across regions, states, and socio-economic groups. The trends mentioned above provide a broad overview of the changes observed at the national level over the last 50 years. What do you mean by hunger and hidden hunger? Comment on various strategies in combating hunger. Hunger refers to a physiological condition caused by the inadequate intake of food, leading to a prolonged lack of essential nutrients and energy. It is characterized by a feeling of discomfort, weakness, and the inability to meet basic dietary needs. Hunger is a severe form of undernutrition and can have significant negative impacts on physical health, cognitive development, and overall well-being. Hidden hunger, on the other hand, refers to a specific form of undernutrition that occurs when individuals lack adequate intake of essential vitamins and minerals, even if they consume enough calories to avoid hunger. Hidden hunger is also known as micronutrient deficiency or malnutrition, as it primarily involves deficiencies of vital micronutrients such as vitamins A, B, C, D, E, and K, and minerals, iron, iodine, zinc, etc. Despite consuming sufficient calories, individuals with hidden hunger lack these essential nutrients, leading to detrimental health effects, compromised immune function, impaired growth and development, and increased vulnerability to diseases. In combating hunger, various strategies and interventions are employed to address both immediate and long-term needs. Some key strategies include 1. Sustainable agriculture and food production. Promoting sustainable agriculture practices that enhance productivity, conserve natural resources, and reduce post-harvest losses is essential in increasing food availability and accessibility. This involves improving agricultural techniques, investing in irrigation systems, promoting climate-resilient farming methods, and supporting smallholder farmers. 2. Food security programs. Implementing food security programs, such as public distribution systems, school meal programs, and targeted nutrition initiatives, helps ensure access to adequate and nutritious food for vulnerable populations. These programs provide essential food support to combat immediate hunger and improve nutritional outcomes. 3. Nutrition education and behavior change. Promoting nutrition education and behavior change programs can help raise awareness about balanced diets, healthy eating habits, and the importance of diverse food sources. This empowers individuals and communities to make informed choices regarding their diets, including the consumption of nutritious foods. 4. Micronutrient supplementation and fortification. Addressing hidden hunger requires targeted interventions, such as micronutrient supplementation and fortification programs. These interventions involve providing vitamin and mineral supplements to vulnerable groups, fortifying staple foods with essential nutrients, and promoting the consumption of fortified food products. 5. Empowering women and girls. Recognizing the critical role of women and girls in food production, nutrition, and household decision-making is crucial. Empowering women through education, skills training, access to resources, and gender equality initiatives can have a positive impact on household food security and nutritional outcomes. 6. Enhanced food systems. Strengthening food systems, including storage, transportation, and distribution networks, helps reduce food waste, improve food quality, and ensure efficient delivery of food from producers to consumers. Investments in infrastructure and logistics can contribute to combating hunger and improving overall food security. 7. Social safety nets and poverty alleviation. Implementing social safety net programs, such as cash transfer schemes, employment programs, and poverty alleviation initiatives, helps address the underlying socio-economic factors contributing to hunger. By reducing poverty and improving income levels, individuals and households can better access food and meet their nutritional needs. 8. Climate change adaptation. Given the growing impacts of climate change on agriculture and food systems, integrating climate change adaptation strategies into food security and nutrition programs is crucial. This involves promoting resilient farming practices, supporting climate-smart agriculture, and developing early warning systems to mitigate the impact of climate-related shocks on food production and availability. Overall, combating hunger requires a multidimensional approach, involving not only addressing immediate food needs but also tackling underlying causes such as poverty, lack of access to resources, and inadequate nutrition. A comprehensive and holistic approach that combines interventions related to agriculture, nutrition, education, and social development is necessary to achieve sustainable solutions and eradicate hunger in all its forms. What are the different national dietary guidelines of India? What is your opinion on balanced diet of Indians? As of my knowledge cut-off in September 2021, the Government of India has not released a specific set of national dietary guidelines. However, there are various recommendations and guidelines related to nutrition and dietary patterns that have been issued by different organizations in India. These guidelines are based on scientific evidence and aim to promote healthy eating habits and balanced nutrition. Some key guidelines and recommendations include 1. Indian Council of Medical Research, ICMR, Dietary Guidelines. The ICMR has published dietary guidelines that emphasize the consumption of a balanced diet with an appropriate mix of macronutrients, carbohydrates, proteins, and fats, adequate intake of fruits and vegetables, limited intake of added sugars, and reduced salt consumption. It also highlights the importance of portion control, regular physical activity, and maintaining a healthy body weight. 2. National Institute of Nutrition, NIN, Recommendations. NIN provides dietary recommendations for different age groups, including infants, children, adolescents, adults, and older adults. These recommendations emphasize the consumption of a variety of foods from different food groups, portion control, and moderation in the intake of fats, sugars, and salt. It also highlights the importance of micronutrients and the inclusion of locally available and traditional foods in the diet. 3. Food and Nutrition Board, FNB, Guidelines. The FNB, under the Ministry of Women and Child Development, has issued guidelines related to nutrition and balanced diets. These guidelines focus on adequate nutrient intake, including vitamins, minerals, and macronutrients, as well as the importance of breastfeeding, appropriate complementary feeding for infants and young children, and the inclusion of diverse food groups in daily meals. Regarding your question about my opinion on the balanced diet of Indians, as an I-language model, I don't have personal opinions. However, I can provide general insights on the topic. A balanced diet for Indians, as recommended by various health authorities, typically emphasizes the consumption of a variety of whole foods, including fruits, vegetables, whole grains, legumes, lean proteins, and healthy fats. It involves limiting the intake of processed and fried foods, sugary beverages, and excessive salt. Portion control, moderation in the consumption of calorie-dense foods, and regular physical activity are also important components of a balanced diet. It is worth noting that individual dietary needs may vary based on factors such as age, gender, activity level, and underlying health conditions. Consulting a healthcare professional or registered dietitian is recommended to personalize dietary recommendations and ensure individual nutritional requirements are met.